All right, we're getting the presentation up, but it's my pleasure to introduce Dr. Anirban Sengupta. Anirban is a research instructor in the Department of Radiology at Vanderbilt University. His research uses structural and functional MRI to probe clinically relevant neuroscience questions in primate models. He's one of the few researchers on the planet with expertise in animal spinal cord functional MRI. So it's a really exciting to have him here. Using animal models, he's revealing how the intrinsic functional networks, both intra and intersegmental connectivities, change in spinal cord injury, providing key insights into the impact of spinal injury on central nervous system and the recovery of sensory and motor function. So thank you for being here, Anirban.
Hi. Yeah.
Awesome.
Can you hear me? Okay. Thanks, Ken, firstly for inviting me for this talk. And yeah, a lot of very nice results that we have seen from the morning on human studies. So this may be a good time to actually talk a little bit about our findings in animals. We do animal studies and human studies also, but I'm right now focusing on the animal studies. We are working at Vanderbilt in Nashville, Tennessee. And yeah, let's go ahead with this talk. So firstly, let me first talk about the motivation. So maybe most of us are aware of this fact that animal studies allows us to implement various experimental conditions in a much more controlled way than humans. So that's one of the primary reasons that many people prefer animal work initially. Also the fact that we can do a lot of injury models and make them similar to those that we see in human also helps in animal studies. Also the fact that we can investigate various drug treatments and see how these can be actually translated to human applications is a big motivation for using animal models. But also I would like to say that many years evolutionary biology questions also comes up because there are the new world species, the old world species, and people always try to see what are the functional homologies and the structural homologies between the different species. So in my study, I'm particularly focusing on squirrel monkeys, which is a new world species. And most of the results, apart from one small result at the end, will focus on the squirrel monkey studies from my group. So the challenges of spinal cord imaging in animal models are very similar to those that we see in humans. Firstly, we all know that the spinal cord is very small in size, that requires very high resolution, which is same in humans. Only the fact that in animals, the spinal cord is even smaller and even higher resolution is required for imaging them. Then the known artifacts that arise because of B0 inhomogeneity, both temporal as well as the static B0 inhomogeneities from the lung vertebra and also from the respiration cycle, cardiac cycle are well documented in spinal cord literature. So these are the things that remain the same in preclinical models too. So let's first start with the acquisition for the structural imaging that we use in squirrel monkeys. We use a very high magnetic field of 9.4 Tesla with a 21 centimeter Bohr magnet. And we primarily focus on the cervical segments between C3 and C7. I will come about how we locate them and things like that later on. For in-plane resolution in the anatomical domain, we go to as high resolution as 0.25 millimeter square based on the size of the spinal cord. And we keep a slice thickness of 3 millimeter. And most importantly, we use a very optimized quadrature spine coil, which is in-house built, which has 3 centimeter width for each loop. So these are the major acquisitional informations for structural imaging. Oh, I forgot. So this is our uh, the acquisition from the different spinal segments, and we acquire this anatomical scans with a resolution of 0.25. And yeah, this is the image for specialized quadrature spinal cord coil. So coming to the structural image, as Julian was discussing yesterday also, we prefer the MTC weighted image because it has been shown that it shows higher contrast between the gray and white matter in the spinal cord. Also, it shows a much higher intensity and specificity towards the injury site compared to the traditional T1 weighted imaging here. So the MTC contrast is the one that we prefer for structural imaging. Now, for the functional imaging, we use fast GRE sequences, as Rob Barry and all, they have shown that it works much better than the EPI data. And we use high in-plane resolution of 0.5 millimeters square, acquiring around 300 dynamics, and also we have a volume acquisition time of 3 seconds from the 5 segments that we imagine. Regarding the coil that we use, as most of us will be aware, that we require a surface kind of a coil because small bore size of the animal scanner. Also, we use a very optimized quadrature coil, which has 2 component coils but a single channel. What it has shown us is that it gives a much better, higher SNR in case of the optimized coil compared to the traditional coil that we used before, which has a single coil, not a quadrature shape. Yeah. So when we talk about animal studies, the first primary difference that we can think of is that we normally have to anesthetize the animals, which we cannot do in human, of course. I don't know if it was done at any point of time or not, but in animals we do anesthetize studies. So one major advantage is that it helps in reducing the motion artifacts by a lot, but it also diminishes bold signal effects. Which has been known. But importantly, what our studies have shown, and this is from brain, but it also, like we assume that it is also true for the spinal cord, is that the suppression effects of the isoflurane, the anesthesia, are not functionally selective. What that means is that if we change the anesthesia between a certain range, the difference that happens because of the different anesthesia levels is not specific to a particular region, but it is similar across the different regions. So this is what we have shown before. Regarding the pre-processing pipeline for our functional MRI, we follow a very similar pipeline to those used in human, as in we used 2D motion correction first, then we go with slice time correction, nuisance signal correction, which involves the motion parameters and non-spine area. We don't do white matter signal regression in our group, and I will show a few interesting results in the later part of the talk as to how that has helped our findings in a different way. We do physiological signal correction using retro-ICR course, and but we don't do the cardiac signal correction, assuming that the cardiac signal has a much higher frequency in case of animal studies. And we do a bandpass filtering, which is in the normal frequency range that we do in humans. We do 2D linear registration. We use FSL, SPM, and sometimes we are also trying to move towards ANTs for this purpose, in which the normal procedure involves first registering the functional imaging data to the subject space first, and then registering the subject data to the template space. And we have an in-house developed template also. And finally, using the transformation matrix from the subject's anatomical space to the template space and applying that to the functional data. So that's the normal pipeline that we follow. A few important things regarding the quality check that we do after our data acquisition or during the processing is that as we were discussing before, the TSNR becomes a very important parameter to check, and especially we do check it for the end slices because that's where the signal drops happen most, because if we don't have a good coil, that can be most affected. Um, and we do check for motion parameters, and if there is motion even after all the anesthesia and other different physical restraints that we apply, then if the motion we find is more than a single voxel's dimension, we normally remove those data from our analysis. One final thing is that we always go for visual check of registration, because as much as these registration softwares give us very good results and optimized results and things like that, it is not perfect 100% even in animals, and I don't think in human also maybe. So we do check for manual registration if that is required after each scan, and do that manually also if that is required. So yeah, these are the motion parameters that I was talking about, and yeah, we do check the manual, the registration, and if required, we do some manual registration too. Now, some of the key things to consider for animal spinal cord fMRI imaging is that we require a very high field magnet. We go for a 9.4 Tesla. This is mostly because we need high SNR to image those small regions, but also because the spatial resolution that we lose, the SNR that we lose because of the high spatial resolution necessity, is kind of compensated by these high field magnets. Also, it is known that as we go towards the higher field, the specificity from microvasculature, where the neural origin is believed to be, is much better at high fields. We have an optimized spinal cord coil, which takes into account the body dimensions of the species, the bore size of the magnet, and this affects the TSNR values, as most of us are aware. We check for the end slice TSNR specifically. Uh, we do a very refined registration, and because slight misalignment can affect our registration, our results in the group studies. So we do manual registration also if required. One tip that I think I would like to give here is that often we have found that cropping or masking or using a Gaussian filter just to focus on the spinal cord part of the image is immensely helpful for the registration results. And I know in the human spinal cord toolbox also they apply a kind of masking for that purpose. So yeah, the registration is something that I would like to give more emphasis for checking during the quality control part of the analysis. Um, the other important things to know here is that anesthesia, if it is used, and in certain studies nowadays in preclinical models, they are also using awake models. Anesthesia has a lot of advantage, but it also requires optimally, the dosage has to be optimized accordingly so that its effects on the bold signal is not specific in particular regions. Also, physically restraining the animal helps a lot in reducing the motion. We are able to do that using placing earbuds. We do headbuds and also elastic straps around the monkey's body while imaging, and this helps in restricting the motion by quite a bit. Also, the important thing is that we do mechanical ventilation. So we actually synchronize the fMRI acquisition with the respiration pattern. And then correcting for these respiration effects later on becomes much easier because they are synchronized with the bold signal also. So these are some of the things that we can do in preclinical models that has been very helpful for us in various ways. The other thing is that we also apply retro-ICR, of course, but the cardiac signal, in case of animal models, we have heartbeats ranging from 250 to 260, which is much higher beats per minute, which is much higher than the frequency range. Also, we have found, and this is something that I know many people have, or they have expressed their ideas about it, that does this correction methods always is helpful or not? Retro-ICOR we do in our studies, but we have also seen that it doesn't improve the connectivity results much in our data. We don't normally do smoothing. One important reason is that because of the small dimensions, we don't want to lose on the signal, especially between the gray and white matter interfaces and those things. So smoothing is something we avoid. We do the anatomical landmark finding using nerve roots, contrasting with vertebra and other things which are used in human. And we use the nerve roots, especially between the C4, C5 segments, sometimes for focusing our or finding out the landmarks in the studies. And this helps in consistent spinal segment-based acquisitions in our study. Also, in many cases in preclinical studies, there is a lack of templates, so we develop our own templates for our purpose. We have a lot of animals in our cohort, so we develop an in-house template, and that helps in the registration process for group studies also. So these are the basic considerations. With this, I would like to go and talk about how we apply our different spinal cord models in healthy animals first, and then we will talk about injury models also. So this was the first study I think I got involved in which we tried to look into the functional networks in the spinal cord. So traditionally, until the last decade also, I think the major focus was on the four horns, the dorsal and the ventral horns, which are known to be the regions through which sensory and motor information transmission happens between the brain and the peripheral part of the body. But as we know that the spine has a much more laminar structure than this, and there have been increasing evidence of specificity with respect to different functional tasks to different regions. So this actually kind of motivated us to study the functional architecture of the spinal cord in much more details. So we used independent component analysis, and this has been shown very effective in brain models before in humans, and we used a data-driven technique, independent component analysis, which basically takes in the spinal cord fMRI data and then gives us intrinsic functional networks on the spinal cord. And I can talk a little more about the mechanisms and all if people are interested at the end of the talk about it. But most importantly, what it helps us is that it does not require any assumption regarding the location of the functional regions in the spinal cord. So we can basically do a data-driven process in case of spinal cord functional network analysis. And what we found using that is that we found 7 distinct regions of synchronous bolt fluctuations from different parts of the gray matter. And as you can see that the first 4 of these are the well documented, the dorsal and the ventral horns from the right and left side. But we also found that there are synchronous activities going on in the intermediate, and we coined the term intermediate because it's between the dorsal and the ventral horn, but to be very accurate, it is more closer to the dorsal horns. And there is also a functional hub that came up in the gray commissure region. So we were very excited with these findings, and we wanted to see it in how it like replicates in human studies. So people who are working on human also in our institute at the same place, they did that in 3T and at 7T in human, not in 7T, sorry, 3T, but in cervical and in the lumbar parts. And they also could find the different 7 locations that we have actually found in preclinical model using ICFS techniques. Now, one of the functional significance of this intermediate region I would like to focus on is that the location of this dorsal horn and the dorsal intermediate region actually matched very well with the known previous tactile and heat stimulation activation maps that we have observed in monkey spinal cord. So the heat activation actually heat stimulation activates the superficial layers of the dorsal horns, whereas the tactile stimulation activates the lower part of the dorsal horn regions, which is just below the superficial layers. And this is exactly where we have actually also obtained the different two functional networks in the monkey spinal cord using resting state data. So this was very kind of promising result in the sense that it also gave us a validation of the fact that resting state networks are actually reflection of the regions which are co-activated during functional tasks, something which people have always talked about in brain literature quite a lot. Yeah.
The tactile, is that noxious or is that innocuous?
Noxious. The motivation behind bold fMRI investigations um, in the white matter, I will talk now about white matter part in the spinal cord, is that so we have seen in the previous literature that white matter signals have been generally regressed out in the brain, and there is a lot of controversy behind the origins of the white matter signal. One of the reasons being that there is reduced blood flow and volume in the white matter and gray matter, as well as there is less energy requirement in the white matter. But on carefully looking in the literature, what we found is that many of these studies have actually discovered white matter, but they have not preferred or conveniently not reported them. Like in this particular study that we saw that there is white matter networks or white matter connectivity observed in the deep corpus callosum region between the two hemispheres, but it was not very much highlighted by the authors. And in our later on, we also found that in using appropriate techniques, we can actually detect white matter signals in the brain of humans, and also we found that the white matter activity was actually anesthesia level dependent, similar to what we see in gray matter. So all of these findings actually motivated us to investigate the bold fMRI signal in spinal cord of monkeys. So using the same technique as we used for the gray matter, using ICA-based techniques, we could find that there was synchronous bold fluctuations observed in the white matter region of the spinal cord, in which we did not regress out, of course, the white matter part of the signal. And the most interesting thing is that this location of this networks was found to coincide very well with the locations that we know traditionally for the anatomical tracts in the spinal cord pathways. For example, we could find locations activated in the ascending tracts, in the lateral tracts, and I would like to refer to, I think, Kinani's group work, Dimitri's work here, that they also had shown that they find that there are certain tracts which are found using their model in the white matter region. So we found very fine-grained neural architecture in the white matter part of the spinal cord using ICA. And this actually matched with the location of the anatomical tracts that we know in humans. Interestingly, we also found that the connectivity, when we did the connectivity analysis, we found that the connectivity is very much highly located along the longitudinal direction, along the tracts, implying that the white matter tracts has a lot of connectivity going on between the different segments, which actually kind of highlight that they serve not only as structural connections, but may have functional relevance too. And motivated by this, we actually thought of moving towards task-based studies to see how that works in task experiments. So we had already shown before that using tactile tactile stimulation, we could actually activate the ipsilateral dorsal horns in the spinal cord gray matter. And this was known, like consistent with the known neural circuitry of sensory pathways. But in this study, we also found that we could find that there is activation in the white matter tracts, and these are very specific to those in the ascending pathways, implying that they have a lot of functional specificity also with related to the locations. And we could see here that the ascending tracts, such as the spinocerebellar tract, the dorsal column tracts, having high activation when we do a simple tactile based stimulus activation on the monkey digits. Yeah. So one of the recent studies that we have just published here is regarding, we were trying to see how the activation models actually work in the spinal cord when we do a top-down approach instead of the traditional natural way of tactile or any other peripheral stimulus-based approach. So what we used is that we used optogenetics to excite or activate the somatosensory S2 neurons in the brain. And we could find out activation in the somatosensory cortex, S2 cortex for that. But more importantly, we could find that there was activity which was going on correlated with the task is in the spinal cord also. And this top-down activation actually provides a new way to integrate the functional connectivity within the brain spinal cord neural circuitry architecture. But also importantly, what we found is that the spinal cord activation included both the gray matter as well as the white matter pathways. So we could find white matter activation both in the natural tactile way of stimulation as well as optogenetically, specifically activating the somatosensory cortex of the brain. One last study that I would like to mention, our group had done quite some time back, but it is of importance here, is that we also wanted to know how much there is correspondence between the functional MRI connectivities that we get using fMRI with those that we can get using electrophysiology, because bold fMRI is after all considered to be an indirect measure of the neural activity. So one of our grad students, he measured intraspinal, he took intraspinal recordings of electrophysiological signals, the local field potential signals, and he observed that there is a very high correspondence between the fMRI connectivity and the LFP-based coherence connectivity between the different regions, which further provides evidence of the neural origins of this bold signal that we are observing in the spinal cord. So with this findings, we would I would move to the last part of my talk, which is on injury models in animals. And we started with a dorsal column lesion model, which is well documented in animal literature. So what we did is that we had transaction of the dorsal column tract at the C5 segment of the spinal cord using scissors, basically fine scissors, and we could see that this was the injury site, and we found that the MTC image that there was change in the in the intensity, signifying the injury effects in the dorsal column region of the spinal cord. And this dorsal column was also chosen because it is known that it is the tract that is responsible for transmitting, or the major tract responsible for transmitting of signals in the ascending pathways. And then what we did is that we acquired spinal cord fMRI data before the injury and also after the injury at various multiple time points. So one of our initial studies found out in this dorsal column lesion model that the injury affects the spinal cord functional connectivity networks, and this is very specific to certain regions. For example, we could see that the injury was more prevalent in the intraslice connections or intrasegmental connections below the injury level. And also there was a drop in the connectivity across the segments on both sides of the injury location. And more importantly, the injury effects were very highly significant in the injured side of the spinal cord compared to the non-injured side. So this kind of gave us a lot of confidence regarding the spatial specificity of bold signals in finding out injury effects after a dorsal column lesion injury in the spinal cord. We also continued the study for a longitudinal period over 6 months, and what we found is that there was first decrease in the connectivity, as you can see, but then there was a kind of a increase back on the connectivity, although it didn't reach the baseline level, but it also signified that there is recovery going on in the connectivity measures across the time period that we looked into. We actually thought of seeing how the communities that we can develop using graph theory models based on these connectivity measures change along these time periods. And we found that there were two major communities. One was the dorsal community, which we are showing here as the second community, and the other community was the formed of the other 5 regions in the gray matter horns. What we found is that the major effect in the community structure was found in the second community, that is the dorsal community, suggesting that although injury effects are altering the functional networks at the different spinal cord levels, but it had a much severe effect on the dorsal column region, which is where we actually injured the monkeys, again suggesting the specificity of the bold signals in capturing this type of injury effects. Another interesting fact that we observed in that paper is that the community structures actually change over the period of time. And here I'm showing the community structure that we got just after the injury at the acute stage. And what we found is that it shows that there is a reduction in the inter-segmental connectivity, whereas there is an increase in the intra-segmental connectivity, showing that the community structures become very localized to within a segment, losing their connectivity across the segment in transmission of neural signal to and fro from the brain. So this was also observed in our study. Now I will be talking about ongoing study, in which we are trying to look into the effects of a motor cortex lesion in the brain of the squirrel monkeys, and want to see how that affects the spinal cord networks. So we already know that the motor cortex has the corticospinal tract that moves downwards and connects and kind of terminates in the ventral horns of the spinal cord. So that was the motivation that we believed that this can actually also, motor cortex lesion can actually impair the spinal cord networks. And we also thought of paralleling them and comparing them with the behavioral functional activities of the monkeys during this period of time. So we looked into till now 9 weeks of the study, and we have continuously acquired behavioral data also along this period and compared them with the fMRI metrics from the spinal cord. And what I'm going to show you is like very interesting result. What we see is that the behavioral measures, as we can see, that they drop down just after the injury, but they also regain back over time. And the different levels, P3, P4, just refer to the different behavioral task models. But what we see in the fMRI connectivity measures is that many of these fMRI measures from the gray and white matter have a very high correspondence to these behavioral measures, behavioral task measures. And this is kind of opens up a new window of looking or inspecting or predicting behavioral trajectory using fMRI and possibly use them as a biomarker for behavioral recovery process in humans also in later. Finally, I would like to talk about another study in the rat model, which another lab member has worked on, and he used rats, and he did a injury in the spinal cord. So he also looked into how the drug treatment actually changes the recovery process after the spinal cord injury. So he used riluzole, which is a FDA-approved drug, and he looked into the behavioral measures. Also here you can see the behavioral measures also, how that is changing with the drug treated group versus a vehicle treated group, which the vehicle treated group is just given a solution but not the actual drug. And then we compared it with the imaging metrics here. And what we found is that there is very high correspondence related to the imaging metrics with the drug treated group versus the non-drug treated group. And this was actually validated also looking into the histopathology staining that we observed after the animals were sacrificed, which actually kind of suggests that imaging biomarkers, imaging matrices can actually serve as biomarkers to injury, but also drug-related recovery processes also. And this can be actually taken forward for translational studies in humans, and that is our next plan in monkey stuff also. Also, we may want to do it at one point. So finally, I will give you a few takeaways from animal spinal cord studies. As Ken has asked also, they'll give some key insights into how animal studies are different and things like that. So in general, the animal studies allows you to investigate various neurobiological questions in a much controlled fashion compared to maybe humans, that we can look into the different functional pathways, pain pathways, and different network organizations. Also, it allows us to do interspecies comparisons and also look into the evolutionary biology studies. For example, in monkeys, we find that there is a lot of connectivity going on between the dorsal and the ventral horns compared to what we see in the humans, maybe related to their activities, which are they use both their hind limbs and forelimbs for their activities. So different species related difference will be reflected in their connectivity matrices also. Also, it allows us to implement various injury models, and we can mimic them with those of the human neuro disorders in the spine, in the brain, or other places also. And we can look into them in a longitudinal study in again a very controlled fashion. So animal studies helps us in various ways in those manner. Some of the key things that I would like to mention for maybe setting up animal spinal cord study labs is to combine the spinal cord MRI with maybe electrophysiology lab, because electrophysiology often validates fMRI as both are related directly or indirectly to neural activity. I mean, electrophysiology is directly related to neural activity, and we can perform intraspinal recordings also in animal models. One important thing is that to use behavioral task related information alongside spinal cord fMRI, because it helps to validate the functional relevance of functional MRI measures, and we have a lot of motor and sensory, somatosensory behavioral tasks models already designed for animal models, which are very well documented. Also, coupling it with a histopathology lab, because histopathology finally gives you the gold standard validation for in vivo imaging biomarkers. So coupling it with the histopathology lab is a very ideal process. All these things you can do in animal studies much easier. And finally, for translational studies, we can actually look into how the treatment based on some pharmacological drug can actually change the spinal cord injury data or spinal cord injury imaging metrics and validate those imaging metrics as biomarkers of response to treatment. So this can be used as for translational models. So these are the various things that we can do with animal studies. And finally, I would like to thank all my group members who have been working on these projects for last 10 or 12 years. I have joined the group in last 6 years, but this is a continuation of work from various other group members. And yeah, thank you all for your attention.
Amazing presentation. Just want to confirm, Mary, are we getting kicked out of here at noon? Or
as far as I know,
as far as I know, we are going to get kicked out of here at noon, and we have to clean up a little bit. So we'll just do quick 5 minutes of questions. Anirban will be on the panel, so we can ask lots of animal questions. I'll start off. I really enjoyed the optogenetic study where you're doing the S1 stimulation and then showing spinal cord activity based off S1 activation, which I think is really neat and kind of shows just how powerful spinal cord functional MRI could be to understand motor control and complex aspects of it, where it's more than just that descending efferent drive to motor neurons. and maybe we need this technology to really disentangle those things and what's happening with motor control. Next, my question is with your thermal and tactile study. Could you just repeat what the stimuli were first?
So these are 8 hertz vibrotactile stimulus. So
Okay.
these are non-noxious.
Non-noxious.
Okay.
Okay.
I think
Yeah,
I missed
think
that.
that was. And
Yeah.
then there's also the thermal one.
I have not shown the thermals, but we
Yeah.
go till 53 degree Fahrenheit. Yeah.
Okay.
Yeah,
So that's
Celsius.
noxious, probably.
Yeah, that may be in the noxious region. But I think in today morning also, Alexander told that heat stimuli is not a very does not generate activations very stable as much as we see in the tactile. Yeah.
Excellent. Any questions?
Thank you for the talk. I'd like to go back to the consideration about including the white matter or not. So the thing with compared to the brain is that the gray matter and the white matter are kind of inverted. So in the spinal cord, the gray matter is inside, and so it needs to be perfused from the outside and also drained towards the outside. So there's a lot of vasculature that penetrates and that goes out of the gray matter. So that's one big difference with the brain. And so I would not be surprised if I would see bold-related signal in the white matter. Because of drainage effect in T2 star weighted images that many people showed. Ravi Menon, for example, showed that a couple of millimeters away, outside of the cortical region, we would actually see some bold related signal. So to me, that's like an incentive for not including the white matter as a regressor. So I just wanted your thoughts on that, and open up for discussion maybe.
Yeah, I think that's an excellent question. That's the question of a classic reviewer, I would say, which comes up in most white matter studies. So we did look into the white matter very over a period of time, 2-3 years, and we focused a lot of analysis on that. And we did. So one suggestion which comes to my mind is that if we regress out the gray matter signal and look specifically into the white matter, just like we do for gray matter, and regress out the white matter, and that can be one way of validating the results and see how the gray matter signal is affecting because of the vasculature and all that we talked about. Also, we found a lot of confidence from Neville's work also, because I think as far as I remember, they also found there are resting state networks in the spinal cord white matter using their ICAP modality, although it was not as fine grained, but they did find lateral tracts and anterior posterior tracts in the white matter of the spinal cord. So there were a lot of considerations on it, and the reviewer, as Rob Berry was also saying, that they will ask a lot of different kind of analysis to verify it. So we did separately masking out gray matter and white matter and looking into the networks, and then also combining the entire spinal cord and looking into those networks. So we did look into one final thing I would like maybe to go ahead also look into is that use much more specific signal from Spinal fMRI, and because they can help to take out the effects from the bigger vessel vasculature and all, and see if that may be helpful in making or convincing people about the white matter signal. But I know this is a very ongoing debate, and people do white matter, some people don't do, but for us the results have shown very high reproducibility, reliability, and also anatomical correspondence, neurobiological correspondence using both resting state data as well as task-based data. Yeah.
All right. Next, any questions?
Thanks. Yeah, nice talk. One of the things we found with doing animal fMRI is the titration between the isoflurane or whatever you're using and motion and the bold signal. And there's a bit of a sharp drop off in the bold signal as you sort of overdo it. And how do you titrate that?
So we know that there is change or alteration in the functional connectivity, and that's also what we show here. What we were more interested is to actually see whether the change is more specific to certain regions or it is not specific to certain regions. And what we found is that within that particular isoflurane range of 0.75 to 1, we find that whatever we see the drop in the bold signal connectivity between regions was not specific to spatial regions, but there is a drop that we know. And most of the animal studies go for isoflurane or any other anesthetic agents for their acquisitions, but they also look into some studies are also going for awake models, and what they have found that it needs a lot of animal training to actually the images using awake animals, and that also doesn't, like, there will be obviously more side effects like motion and all to tackle with, but yeah, anesthesia is a concept that always comes up in the limitation section of our papers, but we also refer to our previous work which shows that anesthesia is much more not functionally selective. Yeah, I would say.
Yeah, okay. We also used training to see.
Okay. Oh, okay.
All right, last question.
I guess go back to the stimuli. So how much is that mapping onto? So I was kind of surprised by the distinction in terms of the laminar organization between the heat and, in your case, the tactile response. And that seemed to switch actually between two of your different studies. One, it was in deep lamina, another is in superficial dorsal horn.
Yeah.
How consistent is that, like, across studies or animals that you guys have? And then how does that map onto kind of, like, we know the electrophysiological profile of the response?
So the physiologically also we know that there is this laminar structure in those part of the dorsal horns, and we did see that there was a finite, very small amount of overlap, but over multiple runs from multiple subjects, and I actually cropped that figure, but there was another part in that figure which shows over multiple subjects. This study was done in 2015, where it shows that there is consistency in that the heat actually activates the superficial layers, whereas the tactile stimulus activates just below the superficial layer in those particular.
So did you guys ever give like a noxious tactile? Because in that case, you should be seeing superficial dorsal horn activation.
I'm not sure about the thresholding, but the overlap between them is very minimal to show. I can go back.
But would that not be surprising if you have a noxious heat? And given what we know from the EPHIS, from monkey literature, you should be getting superficial dorsal horn activation. So would that be surprising in contrast to the two?
Yeah. So I think there is a slight overlap between the first 1 to 3 laminar structures with the fourth to sixth laminar structure between the two activation models. Yeah.
Yeah.
Innocuous tactile.
Yes, this is
right, but yeah, I guess I'm just surprised because we know from the monkey literature that you both have WDRs also in lamina 1. So why aren't you seeing any activation?
I think I'm not aware of that, but I can definitely take this question. Outside this and see and answer it much later. But
I think there should be tactile activation in the more superficial.
In deep, and you're not seeing it like it's lamina 4.
Could you expect up here more?
Yeah.
Okay.
So live corrections?
Yeah, it could be. There could be.
This is
intensity.
Yeah.
Mary, any questions online? Just so we. Okay. All right. So this will end the.
I will end just because I saw there was one question regarding whether we see multiple networks coming up in the same segment or not. So there was this particular, I think, example which often goes in the supplementary section of papers. People don't look into it, but as Dimitri was also talking about that, if we change the number of components in ICA or also in ICAP, I believe, we can actually see dorsal connections bilaterally within the same segment. Whereas if we increase the number of components, we actually see that those components get divided into multiple components. Those networks get divided into multiple components. So this actually shows how the ICA component also influences the network detection models. Yeah.
Awesome. Well, thank you so much. This is fascinating. We're interested in moving into animal MRI as long as I don't handle the animals. So working on it. Lots of great discussions to have later. Everyone, we are
Recording
need to
stop.
clean up, and then we're gonna get kicked out of here. But we still want to take a group photo downstairs in front of the building. Christine's out there with the camera. So let's go find Christine.